Very official. Okay. Well, thank you everyone for being here this evening. I am Sarah Boyce, the Director of Research and Education at the Linda Loring Nature Foundation. Welcome to Science Pub. This is actually the last Science Pub for this year's session, for this year's series. This has been our 8th year of Science Pub. And so I'm really glad to have everyone here that's attending tonight. If this is your first time attending Science Pub, welcome. We have, for the past couple of years, because Science Pub has been virtual and we've recorded our Science Pubs, all of the previous talks are available on our website at llnf.org. We have a diversity of topics and interests and people from all over the country, so we hope that you will check out the previous recordings. So I just have a few program notes before we get started. We have one more event in our community science workshop series. So you're going to be hearing about some community science today. And we had a workshop series this year where we talked about different community science projects that people could be involved in. And the next one is next Thursday, May 19th. I'm going to be hosting it. It's going to be about invasive species mapping and the distribution databases that our state uses. So that will be May 19th at 5:30. It's free. You can register at llnf.org if you're interested. I already said all that. Oh, and one thing I did want to mention, I did mention that this is the final talk in our Science Pub series for this year. We are always looking for additional ideas for topics and speakers for next year. So kind of keep that in mind, and anyone is free to email me. And otherwise, we're going to be sending a short survey to all the Science Pub attendees to ask for some suggestions and things like that. We are recording, as you may have seen when you logged in. And so this lecture will be available sometime in the next couple of days on our website, llnf.org. Just a few logistics. After I introduce Dr. LeBuhn, I'm going to be turning off my screen, but I'll be monitoring the chat and the Q&A. So if you have issues, obviously, with the recording or anything, please let us know. But also, if you have questions as you go along, Gretchen has been very gracious to say that we can interrupt with questions. And then at the end, I can kind of help curate the questions as well. So I think that's all in terms of the logistics stuff, but I am very excited tonight for tonight's Science Pub. We have Dr. Gretchen LeBuhn of San Francisco State University. Gretchen also directs the Great Sunflower Project, a citizen science project designed to identify the pollination deserts across North America and inspire gardeners to do something to help, among many other different research topics. So a pollinator expert on both coasts. So Gretchen, thank you so much for being here today, and I'm very excited to hear your talk, and you can take it away.
All right, great. Thanks, Sarah. I'm so tickled to be here and talking to this group, and I just want to give a plug. I've come to Nantucket for a long time. I have a house on Buzzards Bay, and it is so great to see someone like Sarah running the science at Linda Loring and the amazing work they're doing with restoration. And I really think that properties like that, as many of you know, really provide anchors for the natural diversity that's out there. And you'll see that a little bit in my talk today, though I'll try and shout it out when I get to the points where I can point out how important these natural areas are for providing ecosystem services to the rest of us. All right, let me share my screen and we will get on with talking about community science in the Great Sunflower Project. When I start talking about this, I really like to frame it in that I think community science has changed formal science. And I want to show you today sort of how community scientists are really making a difference for pollinators, and a little bit about the history of community science and sort of just what we know. We're really going to look at 3 different questions today. I always like to start by giving everybody some information to get on the same page. So what do we know about pollination and pollinators? Then I'm going to give a little history of community science and talk about, why we might want to support it and why you might want to get involved. And then I'll wrap up by talking about how you and your garden can help. When we think about pollinators, a lot of people's first reaction is to think about getting stung. But as a scientist who studies the services that pollinators provide to agriculture, to wild areas, this is what I think about. I think about what Bartlett's farm would look like if we actually lost all our pollinators. And if you're a carb avoider, this is like your worst nightmare. Most of the grains that we eat, the grasses, most of the grains we eat, like wheat and rye, those are all grasses and they don't require pollination. But many of the foods that we consume that add the spice in our life and that we can have our children easily say yes to, and that give us the vitamins and minerals that we need, are dependent on pollinators for pollination service. When I talk about pollination service, let me sort of orient you to sort of the what's and whys I'm talking about this. So let's actually start at the end arrow here with the seeds and fruits. If you're an apple farmer, the number of fruits, the number of apples your farm is going to produce really depends on the number of visits by pollinators. It turns out, I think it requires, something like if you're getting 5 to 8 visits in an hour to a particular flower, you're getting enough pollinator visits for full seed set of your apple trees. I'll have to check those numbers. Brian Danforth up at Cornell has been studying this, but it's somewhere around there. So the number of visits that any flower gets determines how many fruits and seeds get produced by that plant. And the number of visits is determined by 2 things. Of course, the abundance of pollinators, but it also turns out that the species of pollinators is important because different pollinators are more or less efficient at pollinating different plants. A good example being that tomato plants have these anthers that only release pollen when they're buzzed at a particular frequency. And it turns out for any of you who are musicians out there, that's about the frequency of a C tuning fork. It's middle C on your piano. And so if a honeybee comes in and tries to pollinate a tomato, they have no effect because they don't buzz at the right frequency. Whereas if a bumblebee comes in, they vibrate at the right frequency, the pollen's released, and that bee is able to move to the next flower and actually do the pollination. All right. So let me give you a guide to our solutions, or a guide to where we're ending up. The bad news that I'm going to tell you about is that bees are declining. The good news is that we don't see big declines yet in fruit and seed set, but we don't know very much, and that's sort of where the great sunflower comes in. And then the last thing I'm going to talk about is really what you can do, which would be one option is the Great Sunflower Project. So I'm a university professor. I like to start with quizzes, and here we go. And perhaps people could put this in the chat. So if I told you that the world had 30,000 species of bees, how many bees do you think there are in Massachusetts? And maybe Sarah, you could shout out any numbers that come in, or give us an estimate of some of the numbers that come in.
Okay, we have 700 is one guess.
Ooh, yeah.
How many bees are there in Massachusetts? I need to hear some more guesses.
All right, I'm going to help you a little bit by telling you that in the United States, there's 4,000.
You could do some quick math and say 4,000 divided by 50. And there's maybe, 900 in per state.
It turns out that there's approximately 390 species in Massachusetts. There was a recent research project done that documented all of the ones in the historical records and all the specimens that had been collected that existed in museums and things like that. And I like to point this out. I like to point out how many there are in Massachusetts because I really think it helps put Nantucket into context. And it also helps you think about honeybees. There's only one species of honeybee. So when we talk about pollinator decline, we're not really just talking about a decline in the number of honeybees. We're talking about declines in the 30,000 species worldwide, the 4,000 in the United States, and the 390 in Massachusetts. How many bees do you think there are in Nantucket, given that there are 390 in Massachusetts? And this number, I don't have an accurate number for Nantucket, but I do for Martha's Vineyard.
Okay, people, I want to hear some guesses of like more or less than, or okay, 200 was one guess.
All right. And we're actually going to give that person a prize because in 2 years of sampling, these two guys, Paul Goldstein, I guess they both have collared shirts, sitting down, and John Asher with the net, surveyed the bees actually out on Martha's Vineyard. The vineyard's bigger, so and it has different types of habitats relative to Nantucket, but it wouldn't be surprising to me if in sampling on Nantucket, we found that there were almost 200 species or around 200 species of bees. Now, if you think about that in terms of diversity, that's a lot of different species. It's really an impressive number for such a small space. And just to give you a vision of what kinds of bees they are, the bees came from 5 different families. The Apidae or the honeybees and bumblebees, Halictidae or sweat bees, Megachilidae or leafcutter bees. Those are the ones that cut circles in your roses. So there was diversity across a number of families. It was not just one type of bee. And it turns out that most of the bees that they found on Nantucket, or rather on the vineyard, nest in the soil. And so when you think about digging up soil on Nantucket, it's really important to think about where you might have nesting bees. It turns out they like sandy, loamy soil. So most of Nantucket is probably good habitat for nesting bees. But they also nest in wood and in the stems of plants. And then if you look at what host plants they use, this is a graph of the different plant families that bees are using. And you can see that this blue area here is the Asteraceae. Those are the daisy-like plants, and that they're really an important resource on all of the Massachusetts islands and Gardner's Island in New York. And then the Ericaceae are the heath plants. Those are the ericas, I can't remember the common name. Those are the different heaths that a lot of the shrubs that you would find out in the areas like around Linda Loring. And another interesting thing about these bees is that they tend not to be found all in the same area. So when these guys sampled out on the vineyard, they looked at how commonly did they find them around 1 or 2 towns, around 3 or 4 towns, around 5 or 6 towns. And they actually found that most bees were only found in one region of the vineyard. So I think that sort of gives you a context that there are a lot of bees out there. They're using a lot of different plant species. Most of them are ground nesters, and we can think about how to do conservation a little bit better once we have things like this in our brain. So when I talk about bees, almost everybody gets the vision of a honeybee in their brain. And I'm just going to blast through a couple of pictures so that you have a visual for why one might fall in love bees. And these are some what I like to call glamour shots that Sam Droege, who works for the U.S. Geological Survey down in Maryland, and one of my students have taken of different bees. So this bee is an agapostman. It's a pretty common garden bee. This is actually very tiny bee. This is a sweat bee. And actually all those gold dots, that's how covered these things get in pollen. This is a habropoda. This is a tropical bee. This is a bumblebee. This is an osmia, a leafcutter bee. And this is oh gosh, I forget which one this is, but you can see sort of how different and varied the different species are. So what do we know about bees? Are there fewer species in areas that are densely populated? Do we get fewer individuals of bees? What else do we see about how bees are put together? Let me bring you back to our map of pollination. So the first thing I want to look at here, I'm going to talk about work that's primarily been done in and around the city of San Francisco, which is where I'm based. But I think the results that we have is really transportable to almost any area. So to look at the number of species of bees, one of my former graduate students, pictured here, Quinn McFrederick, and I, decided to go out and survey the different park areas around San Francisco. And we went out to 20 different parks in the city. They're mapped here. For those of you who know San Francisco, the Golden Gate Bridge would head north here. The Bay Bridge would head this way over to Berkeley. So this goes up to Marin, Napa, and Sonoma. And these are the big city parks that have wildlands on them. And we decided to go just check out those wildlands and see how many different bumblebee species we could find there. Historically, we knew that 9 different species of bumblebee had been recorded in San Francisco. And we were expecting that we would find the majority of them still persisted, though maybe in different proportions. When we went out, we found significantly fewer numbers of bees than we expected. So in black here are the names of the 4 species that we were able to detect. The other 5 species are species that we were unable to find in the city of San Francisco. I've put data from historical records here on the left side to help us show, to help us get a sense of which species were really rare, and maybe we weren't detecting them because they're always rare anyway, and which species were really common. And I like to show this study because until Quinn and I went out and sampled bees, we had no idea that California and San Francisco had lost its most common bumblebee. And if anything is a charismatic megafauna of the insect world, it is bumblebees. And this one, you know, it's a cute little black and gold thing with a little furry white butt. You know, it is as cute as they can come. And even though it was arguably the most common bumblebee in San Francisco, nobody had noticed it disappeared. So this type of story we're unfortunately finding with greater and greater frequency. We've seen the same thing. We've repeated this study looking at solitary bees. We found only 70 species when there are over 150 documented in San Francisco. And so we know that the number of species of bees is in decline in a lot of regions. Well, what about abundance? We also found that there was a much lower abundance of species of bees. And when we looked at abundance, we decided to actually look at what are the factors that seem to predict where there's high and low abundance of bees. And the first factor we looked at was green space. So these are areas like These are parklands, wildlands, these are lawns. And if you look, this graph goes from green space on the bottom here and going from less green space to more green space. And then the number of bees that we found. And you can see that as green space increased, we had an increase in the abundance of bees. We also saw the same sort of thing when we looked at habitat. In areas that had less natural habitat versus more natural habitat, we saw an increase in the number of bees. And this was really one of the first studies to look at this type of relationship. And since this time, there have been numerous studies that have documented exactly the same factors all over the globe. So we see that the abundance of bees has been declining, the number of species has been declining. And the next question is, what does that really mean for the number of seeds and fruits? Well, when we don't have pollinators in the system, we see some really big changes in the number of seeds and fruits. For any of you who have grown strawberries, you might end up from time to time with strawberries that look like these two on the right. This is what strawberries look like when they don't get a visit by pollinators. The difference between these two types of strawberries is one, the one on the far right here, we prevented from having self-pollination and wind pollination. The middle one was allowed to have wind pollination, so pollen grains could have blown on the wind. And the left one here was in open pollination, open pollinated strawberry, where bees, wind, whatever was allowed to pollinate. We also see that the abundance of pollinators, the abundance of fruits declines. So these are data produced by Claire Kremen, looking at the number of cherry tomatoes produced when you prevent pollinators from coming in. That's what the two on the right here, when humans do hand pollination, and then when bees are allowed to do your pollinator, be your pollinator. And you can see that you get a real decline in the number of cherry tomatoes produced when you don't have pollinators in the system. So without pollinators, we get poor quality fruit, and we also get a reduction in the abundance of fruit, or the amount of fruit produced.
Hey, Gretchen,
Yep.
there is a hand raised, and so I'm not sure. So Shirley, if you would like to ask a question, I am going to allow Shirley to talk, if that's okay.
Yeah, let's pause for a second.
Okay. So Shirley, you can go ahead if you want to ask a question. In the meantime, while we're waiting for Shirley, Ken Blackshaw asked, do our bees migrate in and out of Nantucket? This was asked a little bit when we were kind of doing some of the intro to bee work.
No, I don't think there are any bees that migrate. I would be surprised if any bees migrated off of Nantucket. I think that they all overwinter there, and they overwinter. Most bees spend about 48 weeks of the year in their nest, and 2 to 5 weeks flying above ground. And the reason I hesitated when I said that is there are data from a lake in Sweden that showed that bees can actually travel a distance that's greater than the distance from Cape Cod to Nantucket. And so there may be some bumblebees that do come from Cape Cod to Nantucket periodically, but I wouldn't consider them to be migratory. They may just arrive there from time to time.
I think on a day like today where it's very windy,
Yes.
I can consider it episodic. Yeah. So
They have a big influx of different bees today.
So Shirley, if you'd like to ask your question, go ahead. Otherwise, I would ask you to type it in the chat, and then I can ask it later to Gretchen.
All right, do you want me to go, Sarah?
Yeah, go ahead.
All right, so who are the pollinators? Well, it turns out that most gardens, when you have honeybees in the system, are dominated by honeybees. But when people have looked, they found that native bees, the ones that are there naturally, are more than sufficient to provide full pollination services, except in really extreme examples like the Central Valley of California. Here in the city of San Francisco, we've done some work looking at whether you can get full cherry tomato pollination, and absolutely, we don't need any honeybees in our system. So let me just wrap up this part. We find that bees are declining in populated areas and elsewhere, and that they clearly benefit from more habitat. And we do see that native bee pollinator service appears to be adequate. I actually didn't show those data. And we've done some studies in San Francisco that shows that if you have a garden with tomatoes and you add flowers, you have a flower garden with that, you actually get much higher pollination service. So if you want to have good tomatoes this summer, it's a really good thing to have a good pollinator garden too. All right, so let me move on. I'm going to talk a little bit about how your gardens and wild spaces can help, and just sort of what role what is community science, and how it participates. Sorry, I'm tripping over my tongue. It's late in the day. So community science, which sometimes gets called citizen science, I like the definition where we call it scientific research that's conducted in whole or in part by volunteer scientists. And when people have estimated the value of this work to science, we find that volunteers contribute about $2.5 billion in labor annually to biodiversity research. And that's pretty phenomenal. So why might you participate in community science, and why might scientists engage? We're finding that it's an increasingly important tool, and I'll go over some reasons why. The potential of community science really has been dramatically increased by us having better web tools. There's clear recognition from the greater community that if we're going to make informed conservation decisions, we really need scientific data. And I believe that data is really often better than ideology if we're going to come to social consensus around questions of conservation and other difficult issues. So community science has really exploded. And that in part is that we see better web tools. There's been a real push for science literacy. Scientists themselves have really changed their thinking and become much more open to collaboration with non-scientists. And there's a public interest in conservation. The last thing that I think has really driven this is that there's really been some success of key projects. I'm lucky enough to have started a project that has been the model for a number of other projects. And I think that was in part that when we started the Great Sunflower Project, within the first 2 years, we had 40,000 people sign up, which caused all sorts of problems, but really shocked the scientific community that there was that much interest in participating in doing science. Well, it turns out that community science actually isn't new. And if you go back and look in the literature, there's been public participation in gathering data on phenology. There are cherry blossom dates that have been recorded in Japan for over 1,200 years. There have been data on locust outbreaks collected in China for 3,500 years, studying invasive species. And there's been data on species richness. So people have tracked the grape harvest in France for 640 years. So there's lots of examples, if you really look closely, that shows that we shouldn't have been that surprised by the benefits of collaboration between academic or formal scientists and community scientists. What it does do, which is really amazing, is allows us to look at things at much greater geographic scales. If you can only imagine how much it would have cost to send a graduate student out or an undergraduate out to each of these dots on this map to collect data on population trends of wood thrush, it would have broken the budget of any research project. It also has allowed us to look at things across greater temporal scales. So Lilac Watch has been around from, I think, like 1910. So, almost over 100 years collecting data on when lilacs bloom that's given us insights into things like climate change. Community science has improved science's ability to detect disease. Feeder Watch detected this eye disease in house finches, and through their data was able to track sort of how the disease moved across the United States. There's been participation in projects that improve data and image analysis. There's a great community science program out there called Foldit, and Foldit has you try to put together DNA molecules, and they were able to actually construct the correct molecule configuration for an AIDS-like disease in monkeys. And researchers, it had taken them over 13 years to get there. And when they put it out to the Foldit community, the gamers got it in 3 weeks. And then there are things like Galaxy Zoo, where people are making new discoveries. Galaxy Zoo started in 2007. And in the first year, they had 50 million classifications of images from 150,000 volunteers. There have been over 50 papers published with these data, and there actually have been new objects discovered, something called the Hanny's Vorwerp, and there are new types of galaxies that people have discovered. So the contributions to science have been huge, but there also have been real societal benefits. And there's a cottage industry that's sprung up looking at sort of what are the benefits of getting classrooms and people involved in community science. And folks have shown that it improves critical thinking, it improves science learning and bonding, it improves environmental action, it builds social capital, it improves policy. There are lots of big benefits. So with that background, I want to tell you about my community science project, the Great Sunflower Project. So this is a project that I launched back in 2008, and it's really focused on how do we accelerate the pace of pollinator conservation through community science, and in particular, how do we identify those areas where we need to do conservation on pollinators. So I'm going to break this down into 3 things. I'm going to talk a little bit about the science, then I'm going to talk about action, and sort of wrap up with a little bit of hope. If you remember, I started with this figure, and I sort of skipped over the fact that we didn't really have data on the number of visits. So there's lots of studies that have looked at the number of species. There are lots that have looked at abundance. There are quite a few that have looked at seed and fruit production, but there has not been that much documentation of what I call the ecosystem service. How many visits are different plant species getting, and are they sufficient? And if you really think about this, we know that a few species are declining of bees, but we really don't know anything about what the effect of that loss of bees has had on pollinator service. And I would argue that we can't really talk about the magnitude of the crisis or identify how and where to target our conservation measures unless we have a sense of what's happening globally. And nationally. And what we'd really like to have is something like this. We'd like a map that tells us about the weather for bees. So imagine if we knew that all the red areas here were areas that had sufficient pollinators and the blue areas did not. We could say we need to do pollinator conservation up in this area. So a map is really a critical step towards doing conservation. So the Great Sunflower Project was started with the goal to do this, to create that map, to identify the areas with poor pollinator service. We began with what we called the Lemon Queen Sunflower Program. This is where everybody planted the same variety of sunflower, the Lemon Queen variety. And we've had people participate in well over 1,700 sites across the United States. We have over 26,000 counts that have been made. And we've really been able to sort of build that map. We've got data all across. So we've got participation from all across the United States. Not everybody contributes data. And if you're someone who's not interested in contributing data, but want to hear about pollinator conservation or do some assessment of your garden, we are delighted to have you participate. So the basics of it are this. A participant joins. This is data from Dolores back from 2009. She told me what date she participated. She said she watched one sunflower. It was a lemon queen sunflower. She allowed her phone to geolocate, so I know the latitude and longitude. And she watched that sunflower for 15 minutes and wrote down everything that came in. So she recorded 3 bumblebees and 1 bee that she didn't know. And what I do is I convert that to bees per hour per flower. You can think about that as like the bee speed limit. She also happened to send in a voucher. So I was able to verify that she really was seeing bumblebees. Well, where this data gets amazing is when you can put it into a geographic context. And so I can take her data, draw a circle around the site where she is, and start to know something about what's happening in her region. And as you can see from this map, she lives in a rural area. There are some roads, but not a lot of roads. And I can quantify the crops that are grown there, how much open space there is, how much wild land there is, and really start to understand sort of why her data looks like it does. Well, we've gotten data from lots of different places across the country. And we've started to do some analysis, and I'm just going to show you the East Coast and West Coast work. So this is something called hotspot analysis that identifies areas that have higher and lower than expected pollination service. So everywhere that you see a red or an orange dot is an area that has good pollination service, and everywhere that you see a blue or a green has much lower pollination service. The size of the bubble indicates sort of sites that are similar aggregate into a bubble that's of the same size. So when we piece this out and do some analysis, we actually find that the northeastern United States has higher than expected pollination service. The Northeast seems to be doing pretty well as a whole relative to places like this central below the Great Lakes region of the United States. We also see in California that the Central Valley and the desert regions are doing particularly poorly. When we take these data, we can start to look at things like, how do pesticides affect the pollination service? How does drought affect them? And I'll just show you some drought data from a few years ago to give you a sense of it. So here are individual points. Here you can see the blue ones. I, of course, changed the color scheme when I did this analysis. Blue means good here, red means poor, yellow means that they were around average. And you can see that the Northeast is doing well. They, the blue sort of shading color indicates that that was the 5 years previous were wetter than average. And you can see that the orange areas here were in a 5-year drought, and that drought clearly has an impact on pollination service across the United States. I'm just going to pause here to say that just getting to this map was a success for the Great Sunflower Project. In looking at these maps, these are some of the first maps we had that allowed us to understand what was happening. We've been able to identify areas of high and low pollination service. We can spell out particular region, and we've started to be able to understand what's really happening or what factors influence the ecosystem service of pollinators. All right, I'm going to take a pause for a second check with Sarah, see if there are any questions, and move on to action.
We are okay right now. We don't have any questions right now. I think everyone's saving them to the end.
That's all right. So the next thing that happened in the Great Sunflower Project is we had some people panic. Those folks who discovered that they had no bees, it is late in the day, really wanted to help. And I can't tell you how many emails I got from people who were very concerned when they detected no pollinators on their plants. This led to us collaborating with our participants to develop what we call the Great Habitat Challenge. And the key focus here was asking the question, what did bees need to survive? We focused primarily on urban environments when we came up with our checklist, but it's really useful anywhere that you want to implement it. When we looked at the data, there were lots of suggestions that people were making about how to improve your garden for pollinators, but very little of it was backed up with actual scientific evidence that showed that the things we were suggesting worked. So people suggested you needed to have a water element, that having nesting resources, some bare ground was important, converting over to native plants. So we put together a program that's a checklist, and it has a set of questions focused on trying to figure out what people have in their gardens and finding out what things are actually working. So if you do the Great Habitat Challenge, you'll be asked things like, at your site, are there areas which have with bare ground and very few plants? Because most bees won't dig through a lot to get to the bare ground. Or are you at your sites, are there areas with sandy, loamy soil? And at your site, are there areas with no plowing or tilling? So you can run through the checklist, and at the end of the checklist, we'll give you a suggestion of what you can do to improve your habitat. We were interested in this both from the standpoint of getting the data on which things actually seem to be working, but also to see if a simple tool like this actually leads to conservation changes. When we kept track of how many people who participated actually during the next time they filled out the checklist, took action to improve pollinator habitat, we found that over 70% of the people who simply ran through our Habitat Challenge checklist made a change. And I think this is really great news. It shows that gardeners will engage in conservation actions. We've had over 2,000 sites evaluated. We found that understanding people's perceptions when we designed the project really improved our success. And I wanted to mention in here, we also looked at whether it changed people's attitudes about their ability to do conservation. And we found that it had no impact on that, which made us realize that we're really still preaching to the choir. And so one of the things that we're trying to work on these days is how do we get people who are not passionate about these issues to get involved in doing conservation and participating in community science, which actually brings me to my last part, which is talking about hope. We've had, as a scientist, when I started this, my main goal was really to enhance scientific knowledge and publish papers and learn about, what are the factors influencing conservation. But very quickly, it came to me that in building a community science program, what you're really building is community. And we've had a huge number of people participate. And I just want to draw attention to a couple. We've had a lot of seniors participate. And one of the most touching things that I ever received in my whole life was a letter from a woman who wrote me and said, I've been a lifelong volunteer and I now live in a senior community in North Carolina, and I'm in a wheelchair. And I really thought my years of volunteering were done. But I convinced the gardeners to plant some lemon queen sunflowers, and I have been reporting data every day for your Great Sunflower Project. And you've given back to me a way to give back to the community that I thought was gone from my life. And you get a letter like that, and it changes your life as a scientist. You realize that these interactions and the interest of the community really may be the way that we can change the world. On the other end, I got this letter from a group of kids in Florida, and I just have to share a little bit about it. They wrote me and said, "We are a fourth grade class of gifted students. We grew tons of sunflowers. We're from St. Petersburg, Florida. Our school is Perkins Elementary. We just harvested our sunflower heads, and we've been counting today to see how many seeds are on each head. On one, there were 2 flowers that were sort of Siamese. We're going to send it to you," which they did. "And all around, there were 1,051 seeds on that flower. We have questions." So the first question they asked was, "We know that more than 5 bees came to that flower in less than 30 minutes. We've had lots of bee activity. How do we know if all the seeds we counted are pollinated?" Then they asked, "If there is a seed, does it mean that it was pollinated, or could it be a dud?" So with that, I was able to talk about plant reproduction. I was able to talk about sort of plant-animal interactions. They really got engaged. They asked, "The black sunflower seeds look exactly alike. How do you tell if they're pollinated?" But my favorite question they asked was, "Do bees leave footprints on flowers?" And wouldn't you know, that same week in the top science journal, there was a paper that had come out that showed that when a bumblebee visits a flower, it actually leaves behind a tiny little scent that other bumblebees can detect. And I don't know, I think that's actually a bumblebee footprint. So I was able to talk to these kids at school, tell them that they, in the same moment that science had come out with a paper, they'd had the same question, they'd become scientists. And as someone who's always concerned about science literacy, how do we engage students? I think that participating in projects like this is really the entryway into being engaged citizens with strong scientific literacy and caring about the world that's out there. So I'll wrap up just by saying that I really believe that finding a way to track and value what nature provides, doing things like getting that bee count so you know how many visits your plants are getting, is a way that we're going to find a future where conservation is mainstream, it's economically attractive because it's understood, and it's therefore commonplace throughout the world. So please join us, and here's the link to the Great Sunflower Project. Thank you.
Thank you, Gretchen, so much. I mean, this is the hard part about Science Pub being on Zoom. You don't get to hear all the clapping and thank you, but I especially want to thank you for ending on those beautiful, hopeful, inspiring discussions. I love, here, some of the work we do at Linda Loring, and we have the research side, but we try to connect everything we do with education in some way. And you're absolutely right. The questions that student groups and kids come up with are really amazing because they are often really similar to what people are actively studying. And I love when they get to the boundary of current knowledge, right? And you're like, we don't know, we're still trying to figure that out. That's always amazing to me. Thank you. Gretchen, did you want to share the video or do you want to just?
People are welcome to look at it on their own. If we run out of time, we can share or whatever. Okay,
so just in case you're kind of thinking of signing off soon, we're going to get to some questions, but Gretchen did put in the chat a link, which is a great video about some of the education work at a school in Oakland. Great, I'm gonna check that out. One question that Barbara had was about deer and sunflowers. Do deer eat the sunflowers?
Absolutely, deer eat sunflowers.
Yeah.
Sunflowers are interesting. You do have to protect them, and but I think they're worth protecting because they do a couple different things. It turns out that they're often used in remediation of like chemical nasty chemicals from soil. It turns out that they suck them up. So if you ever have concerns about what's in your soil, sunflowers are really great for that. They provide lots of floral and nectar resources for pollinators. But if you can leave the seed heads there for long enough, they're really an awesome resource, particularly for migratory birds who are coming back through.
Excellent. I wanted to also give a plug for the Nantucket Land Council. If you are on Nantucket, they have started a Planting for Pollinators program. It reminded me, I was reminded of that, Gretchen, when you were talking about the Habitat Challenge. So if you go to the Land Council's website, they do have a place you can sign up for free to register. And they have a great pamphlet that they just put out that recommends for different sizes of yard and different size of garden space, native and some garden varieties that help the local pollinators as well. So I just wanted to give the local shout out for a similar habitat project. We do have a comment, so thank you so much. This was really cool. I'm a student at Bard College in upstate New York, and we're actually trying to get a cornfield converted to sustainable agriculture. Potentially cover crops and sunflowers are in the works. Having the citizen science aspect is great.
Let me respond to that by saying the methods in the Great Sunflower Project, where you observe a plant for a period of time. So you record the time, how many flowers you're watching and how many visitors you got, can really be applied anywhere. So I could imagine you could do a Nantucket wide project where you have people look at different plants that get planted in gardens, different plants in a habitat like at Linda Loring, to see which are the critical resources for your specific area, or even in a community garden or converting in sustainable agriculture. We've also worked with some farmers where they've been interested in whether or not their crops are being limited by pollinators, and they do exactly the same thing. They do timed counts on their apple trees. Are they, getting the same number of pollinators this year as they got last year, as they got 10 years ago? Building the data sets just using this simple protocol really is a powerful way to understand what's happening and watch change through time and across space.
I think that's great. I mean, we participate. I mentioned at the kind of top of the talk about some of the community science workshops that we've given, and it's basically been things that we've already used, right? So National Phenology Network, looking at invasive species, iNaturalists. But I'm really interested in delving into the Great Sunflower Project, because I think here using those protocols here at Linda Loring would be a great idea, because we could either have the native black cherries, which we already monitor for phenology. If we have a few that we tag specifically for this project, you can visit the same one every year and have student groups look and see how the pollinators might be changing. That's exciting.
The LA County Museum has a pollinator walk where they have signs that identify the plant species, and they have them enter data into the Great Sunflower Project at that sign, so that they can track sort of what's happening. And they get data on a day-to-day basis, and they can do things with it on seasonality, all sorts of, which plants are in bloom at what time and how many visits are they getting? Should we switch it? It's really a fun project.
So actually, because you didn't delve into the methods of the Great Sunflower Project as much, do you mind if I ask you a few questions?
No,
I'm monitoring the chat at the same time, but I'm going to selfishly ask my own questions if they're. Please ask, so people who are still on, please continue to ask questions, but I'm going to keep Gretchen here asking questions if I don't see new things come into the chat. So what I wanted to ask was, because it is the Great Sunflower Project, right? But you've mentioned a few times that you could use the same methods on other species. Does the Great Sunflower Project take data on other species?
Oh yeah,
or
sorry.
is that something that we could, like, if I would just collect that data?
So we collect data, and I should have mentioned this, after about 4 years, we had so many people want to give us data because they had a plant that attracted so many bees in their yard. We collect data from all species these days. And it's really great because we're now getting the point that we're building up enough data on different plants that we can do the same type of analyses that we've done using the particular sunflower that we did. So it also means that and I haven't looked at our Massachusetts data, but I could go look and tell you what are the what plants we're getting the most data on in Massachusetts, which ones get the most visits, which ones down on coastal are the ones on coastal Massachusetts the same as inland, you know, all sorts of things like that. So yeah, so it's been really exciting to get data on all sorts of different plant species.
That's exciting. I think I know in my own yard, I plant sunflowers every year, lots of variety of seeds, and they get so tall, and then a deer comes in, or I try to protect them. And so I am conscious of that for others as well.
Yes.
When you were showing that map of the different, like some of the summary data from the Great Sunflower Project, how long was it, or how much data did you have before you were able to do some of that? I'm curious how much. You said 2007 is when you started really the effort of the project.
Yeah, we pretty quickly got enough data to do an analysis. So I'll answer that in different ways. One reason that we have a gap in the center of the country is we felt uncomfortable that that we had enough data to detect trends over time in those areas. So it in some ways depends on what question you're asking. You know, I would say for detecting trend, you know, it's sort of every 5 to 7 years we have enough data that we feel comfortable. And it also depends on use. Because we're a community science project, we find that we have a lot more data in cities than we do in rural areas. And so we've had to sort of wait for rural areas to catch up before doing tons of stuff with the data.
And a follow-up question, do you feel, or have you noticed how COVID stay-at-home orders and people doing a lot more homeschooling, has that changed any of the data acquisition? Because I know that something that we did in our family was like a lot more community science projects.
For funny things, I haven't looked, and I think that's in part that I had kids at home during COVID, and I'm just recovering. But it's a great thing to think about. We do know that we get bumps up when we get national coverage. So like Nature did a special that was the Great Sunflower Project was part of, and we saw a big increase that year. So yes. So there are lots of different things that influence how much we're getting.
That's great. And then my last question, I'm interested because I've worked with different community science projects in the past before my current position. I'm curious about the longevity of the Great Sunflower Project, and kudos to being able to kind of keep it going. And has it been institutionalized, or how has it been keeping going? Are you applying for grants constantly, or I'm just curious how that works?
So we have, we apply for grants, we get, we're actually funded often by private donations. And we run on a shoestring most of the time. And so really, I maintain it. I make sure that the Great Sunflower Project can run even if we have no money for a year. And like right now, we're about to start doing some fundraising because we need to do a real upgrade to the website. And so I will do bigger episodic fundraising. And, you know, we need $15,000 right now, you know, to do what we need to do.
Yeah.
which is not an unattainable amount of money for us, but it means that we start focusing on fundraising again. We have considered, there are a number of citizen science projects that give people the option of joining for $15. You can join and not pay, or join and pay $15. And even having people give $15 when they sign up is enough to generate enough money for us to do magical things. And so we've been debating whether to do that. And we're going to run a little project with the business school here to see sort of what the costs and benefits of that might be. I've been really trying not to have any barriers to participation, but I also know that we have people who, when we ask, are happy to sort of give donations. But as you know, raising money takes a lot of time and effort for the staff. And our staff are me, and then I have a couple people who work part-time when I need them. And so it takes a lot for us to go back and really do a lot of fundraising.
Well, it's good to hear. I think that it's a nice model to, like, not just the community science perspective, the research perspective, the type of data you're collecting. It's like also the model for fundraising. It's like the whole package could be a great model. Another question came in. So this is from Alicia. I have seen an impact on commercial mosquito spray programs in my neighborhood in New Jersey. How can we get more regulation of these pesticide spray programs? They are using neonics sprays that harm bees. Sorry to scroll down. We had many solitary bees in our yard, and 3 years after Mosquito Squad being employed by neighbors, we had none. I've seen that on Nantucket too, in particular yards.
Yes. So I don't know how we can regulate. Well, I think it means you need to get local, regional, state regulations in place. We've just done that in California for neonicotinoids. It passed in the last month. But there's some organizations in particular, the Xerces Society, which is XERCES, has really taken a lead on developing materials to present on the impacts of neonicotinoids, and I'm sure that they would be good partners for helping with that fight. Neonicotinoids clearly have a huge impact on pollinators and other insects, and I would like nothing better than to see a replacement.
Great. All right. Well, I want to thank you again, Gretchen, for being here tonight. This has been really great. I've learned a ton, and I'm really inspired to start some pollinator observations at the Linda Loring Nature Foundation. Hopefully next time you come to Nantucket, we'll have you back out here and we get to chat more about pollinators and pollinator.
Of course.
Diversity.
I'm looking forward to it, Sarah. Thank you so much for everybody for coming, and it's a pleasure to be talking to Nantucket tonight.
Great. All right. Thanks again. I'm going to